try to keep the things simple, right? So here we are in second week. That's what we achieved in week one, and this is how many? 5 weeks training, and every day, like Monday and Tuesday, 2 hours a day, and 2 days a week. That's 4 hours a week. So 5 weeks multiplied by 4, that's equal to 20 hours. So if needed, we can extend beyond 20 hours, but I believe that we're going to finish our targets or our objectives within the given time. Right, so we talked about the NGS overview and applications. We gave you the course overview. We talked about introduction to NGS and Sanger versus NGS, a comparative. And then we were here, applications of NGS. We went through some databases uh, on these slides, and where we talked about different links where you guys can go and click and read about them, where you can get that sequencing data, especially this NGS data. Some of them are generic, so you can have protein data and you can have both RNA and DNA data for the nucleic acids, right? But this course is all about nucleic acids, DNA and RNA sequencing, right? We talked about Sanger sequencing. We started with the DNA, and then we gave the concepts of nucleotides and the difference between the main molecule, that 5 carbon sugar molecule, ribose and deoxyribose. Um, One is in RNA, that is ribose, and deoxyribose is in DNA. We talked about nitrogenous bases, then we talked about that this Watson and Crick double strand of DNA, and we realized that if you have one side of the strand, you can get the other sides of the strand. So that thing, along with the other inventions or discoveries, rather, of the DNA polymerases and other enzymes, and actually that process of replication was in his mind when Sanger did this sequencing. So initially it got famous as DNA sequencing or Sanger sequencing, but later on we started calling it as first generation sequencing because we realized that the second generation came out that got famous as next generation sequencing or NGS. But rather than calling the previous as previous generation, we then started calling them as that this NGS is the second one, and then Sanger was the first right, or first generation sequencing, because now we see there is this third generation of sequencers, right? The way you have different generations of your mobiles or your laptops or some other electronic appliances or computing appliances. Similarly, we had the generation of technologies coming in here in sequencing thing, right? So first gen is Sanger, and we talked about the dideoxy terminator method that is utilized in Sanger sequencing, right? And then we talked about the second or next. So it's just like Sanger, but the difference is that we have different methods for sequencing, and we can process a huge amount of data together. Here on this slide we covered, and these technologies, Illumina and OnTorrent and 454 and ABI machines, are, we call them as NGS or next generation sequencing, right? But the ones like the Sanger, that was the first generation sequencing. And then now we see the third generation, that is Helicose and Oxford Nanopore. We talked about them also during the previous lecture. Then in the library preps, what we discussed was like you break the DNA sequence into small fragments because you cannot process your huge chromosomes or DNA sequences, the longer ones, in those NGS machines. So we break them into short fragments and we sequence them in what we call it as massively parallel sequencing, during which we have those billions of fragments we put together in parallel, so that's why it's called as massively parallel sequencing. This here on this slide we talked about the different instruments on which that sequencing thing is done. Illumina, for example, the Illumina company came up with the flow cell, same way Solid came up with flow chip, and 454 came up with Pico tighter plates, PTPs, right? You want me to make it bigger? I can do that. So right now I'm on this slide because I don't want to spend more time on it because we already covered that 2 times, and this is the just revision we are giving to this because it's been a week, so I believe most of you have forgotten, right? So that's why we are just giving you a revision. So let me have this swap display, right? Now it's better. So flow cell, I was here, and then Solid as flow chip, and PTPs are pico-tighter plates in 454, and Iron Torrent has those iron semiconductor chips. These are the instruments on which that sequencing thing happens, right? Yeah, switch off your mic, please. And the next one is this. On this slide, we covered that these are the different layers of actions we perform here in this. And these are how many? 6 layers, starting with experimentation and library preps. Let me take this thing on the other side. How can I get rid of that? Maybe just tape, and I can go here. Not now. And setup, and I can go with individual window or kiosk. Right, so I think that's better. Yeah, so much better. Yeah, now my other screen is also available, so I don't need to worry about. Even I can make it smaller. Uh, yeah, so I guess let me get rid of this thing and this thing and this thing. I've opened up so many windows here. Right, so yeah, that's fine too. All right, so that's what it is. So we have this experimentation and then extraction of DNA or RNA in case of an RNA sequencing project. We realize that RNA cannot be sequenced directly onto the machine, so we need to convert that into the cDNA. So in the end, it's the DNA that's processed. So we do library preps. We break those DNA fragments or cDNA fragments into further small fragments, right? And I gave you the reason why this thing was called as library preparation. And we talked about the cloning vector libraries that was used in Human Genome Project. We gave you the paper of Human Genome Project. So if you have seen that paper over there, he's discussing different cloning vector libraries because at that time the technology was not that advanced. So we had first generation sequencers. So what they used to do was those cloning vectors to store those larger pieces of the DNA for some time, and then when they were to pull through those sequencer lines, then they used to bring them up from those cloning vectors, right? And those cloning vectors are where you store the DNA and you clone it, you make its copies for future use, right? So since you had only one copy, but while you are processing, you need to process it multiple times. So you won't be able to take those multiple copies, right? So that's why those cloning vectors they were utilized over there. That cloning vector library gave this name library preps, but here you don't need to do that cloning vector thing. You can directly sequence a sample. You get the blood sample, or you get the soil sample, or maybe it's your gut microbiome. Maybe you can get that gut samples. You can get the samples from any body secretions and give it to the sequencer, so it will sequence it directly. And then during the sequencing, there is another layer here that's called as PCR amplification. So that is not the amplification of that DNA or RNA, that starting material. This PCR amplification is those fragments which we already had. They had gone through that library prep, right? So we need to amplify them, we need to increase their number, because when we sequence them, they would give us a light signal. So if it's only one fragment, so obviously that will be a very, very tiny little light. So it will be hard to be detected by these sequencers, because these sequencers, these NGS sequencers, perform sequencing with the process we call it as sequencing by synthesis, SBS. Similarly, Sanger was also doing that, SBS. In fact, Sanger was doing SBS, so they kept it the same way, except the processing speed is increased, right? So that sequencing by synthesis is where you make, you make that DNA a single stranded, and you synthesize or make the other part that is missing here. So that is synthesis. So that's why this thing is called as sequencing by synthesis, right? So in those sequencing by synthesis reactions, as the nucleotides are incorporated, the light signal comes out. That light signal is detected by digital cameras. But if it's only one fragment, and if it's only one nucleotide is being added to any base position of a fragment, so that might be requiring a very, very high resolution camera. So obviously that will be increasing the price of, number one, maybe it's not possible to go up till angstrom level or nanometer level. So obviously they decided to, rather than working on that uh, camera side, they decided to amplify the fragments themselves. So these are the fragments here, these processed fragments to that library prep thing where you did their breaking and then you attach the adapters to them. Those are amplified here in this layer, and then this sequencing layer. And here we observed that there was emulsion PCR in these three technologies, and there was this solid phase or bridge amplification in Illumina, and that was happening on that sequencing instrument, that flow cell. So that's why and that looked like a glass slide, but obviously it's a glass slide bigger than the glass slides which you have been processing through your microscopes. So that's why it's called as PCR on slide. And then since those fragments are being amplified, they make the shapes like bridges. So that's why it's called as bridge amplification. Emulsion mean in an emulsion oil, wet PCR. So that's actually dry, so that's wet. So you have that emulsion oil here, and then you take water droplets within that emulsion oil and you do it there. We discuss them in the next slides. And in sequencing, then in this fifth layer is sequencing. So where ion semiconductor sequencing is done in 9 torrent machines, and 454 or pyrosequencing machine method is adopted by 454, or what was that, the Roche, the company's name was Roche, and this technology got famous as 454. So that's called as 454 pyrosequencing or just pyrosequencing. Then sequencing by ligation, that's performed in solid. And Illumina, there is this reversible terminator technology. So we use the terminators similar to those dideoxy chain terminators in Sanger, but here these terminators are reversible. Sanger terminators are not reversible, right? So that's why it's called as reversible terminator sequencing, right? And this last layer, when you get that sequencing data out, so that's called as data analysis. This course is all about data analysis, right? So yeah, so here in library preps, we discovered that those fragments, since they are in millions, so you probably need to design million different primers for sequencing, but that's what is the downside. So we cannot do that. So rather than that, we may attach a piece of DNA, which we already know, we call it as adapter, right, to the left and right hand sides. Slides are not moving. I think they're moving. All right, and then these adapters are attached to it just to keep track of the forward and reverse primers or forward and reverse adapters. We labeled them in green and red. If anyone is not able to recognize green and red, let us know, because that's the problem with the color blind people. That's what we realized here, and that's what we had this thing here. So but that's how they were depicting them in the literature, right? So there is one downside of it is maybe the people who are colorblind, they might have difficulty in, you know, distinguishing between different colors, right? So once those fragments are you know, they are they are attached, the adapters on both sides, those are the selected fragments. The ones which are missing on either left side or right side, they are the ones that are rejected fragments. They are few, but mostly they are having both the ends attached to those adapters, right? So and then they are made that you know, they are converted into the templates are single stranded entities. So those templates, they move on to the sequencers instruments. Those flow cells are those flow chips, and then over there this sequencing thing is performed. RNA libraries are different because those you need to convert them into the cDNA, right? So those cDNA fragments are then processed, and rest of the party is the same. In paired end sequencing, we take only ends of the target sequencing, right? Sequence, right? So we decide that we're gonna sequence this piece from here to here, and then we attach those adapters to the ends, right? And these adapters are complementary to each other. So we allow them to ligate with each other, and then this thing is circularized. So this is something like you tie a knot to a long thread, right? And then you cut it into pieces, and you only retain the pieces that has this knot thing. Rest of them are then rejected, and you pick up that piece, and then you uh, put the adapters, and rest of the party is the same. So while they go through Illumina sequencer, they are amplified over there. So that's what is happening to have this uh, adapter, or that thing is complementary to this primer here. This purple thing is complementary to that blue thing, and then they get together, they get attached, and then they are duplicated, and then they are, you know, it's a thermocycler going on in between. So that is changing the temperature real quick, giving them the optimal conditions to attach and detach and grow and, you know, separate, right? So in this way you get those bridges, they are formed. We shared videos of all this. I hope you might have seen those videos. So they had made your concepts very clear. So once that fragments are multiplied, we get what we call them as clusters. So each cluster represent one fragment and its multiple copies. So if it's only one fragment, and it should be that, that is the ideal scenario. That's called as monoclonal amplification, right? These are the copies of one fragment, right? If the two fragments, they just stay together at somehow, at, you know, they separated, but they did not occupy the space far off from each other. So if their clusters are mixed with each other, they are not monoclonally amplified, so they will give the wrong message to sequencers, so that sequencing quality will be then decreased, right? So that's what happens when multiple copies of each fragment is formed, when these clusters are formed. So that's cluster generation. And there we are just picking up one of them from each cluster, but trust me, it's happening to all those fragments at the same time concurrently. That there is these primers, the other primers, we call them as sequencing primers. They are attached here, and then those nucleotides, they keep on coming and, you know, attaching, and they are giving us the light signals. And that nucleotides which are attaching are based on the reversible terminators, right? This is what happens in emulsion PCR. So in emulsion PCR, what happens here is, let me bring in another participant. So what happens in emulsion PCR is that you get those fragments, and you take those beads, what we call them as streptavidin beads, and these beads are then attached to the fragments, and those fragments are then multiplied there. An ideal situation is you take that oil and you put water in it. So it's water in oil, and then you shake them till you get those water forming those droplets, and you also put those beads in it and you shake them such that ideal situation is where we want there should be one droplet carrying only one bead, carrying only one fragment here. Why not two fragments? Because then it will not be monoclonally amplified. So in that case, that will give us the wrong signal, or that will give us the wrong sequence, right? So over there will be those errors, or there will be that sequencing quality that will be gone below, right? So here we get the monoclonally amplified balls, right? Those beads. So that has the multiple copies of the same fragments. Again, how many? They said between 10,000 to 20,000 copies. Those that much PCR reactions they should be happen in those cycles. You can that calculate like how many cycles are there, right? Because every time the sequences they multiply, they duplicate, right? And they grow in real quick fashion, and you get those balls that are having those multiple copies of the fragments, and you take those balls or the beads and you put them into that. This is we are seeing that picotitor plate in Roche or in 454. So whether it's PTP or whether it's OIN semiconductor chip, in OIN Turret technology, we are ignoring ABI solid because that's sort of a history lesson. It's gone, not that utilized, though earlier when it came out, that was the most successful platform, right? But 454 or OIN Torrent or Illumina is, I would say, in my view, it's number one, and number two is, I guess, 454 or even OIN Torrent is equally popular, right? So that's where you pack that material into those sequencing instruments, be it PTPs or be it flow cells or flow chips or on semiconductor chips, right? So these are the terminators. We talked about them, the reversible terminators. So that has that molecule on that base position 1 where that nitrogenous base is attached. So we have that whole thing, that whole group, which is called as fluorophore, is attached to nitrogenous base, right? So when that fluorophore gives us the light signal, that thing is detached from somewhere from here, right? Yeah, here. So and then we get this thing, and finally that light signal is gone. And what's happening at the 3' end here, unlike that dideoxy chain terminators, we are not taking its oxygen out, but hydrogen was replaced by nitrogen. So then there is water in the mixture, in the medium, right? So that after that fluorophore signal is detected, that light signal comes out, that hydrogen comes back through a reaction between water and this part, this N3, and um, this thing is then ready to accept the next nucleotide. So that termination is temporary. It's not the permanent terminator, but rather than we call it as reversible terminators. That's what happens in the sequencing instruments in case of Illumina. So each fragment is incorporating a different nucleotide, and that is giving us a different color. So the nucleotide is added, color signal is generated until the sequencer reads that, it stays there, and then that reversible terminator thing comes back, right? It brings back that hydrogen thing, and that 3' OH position is available. So then next nucleotide adds, but in between you get a time where you can detect what has been incorporated. So here is just like your eye is camera in a sequencer, and these are the different positions or different fragments, right? So we are looking at them, and if you see green color, it's a C here, T means red, blue means A, and yellow means G, right? So what do you see in here at time point 1 in this fragment? So total we are seeing here like 20, I guess around 16 or 17 fragments here on all those. And these are the same spots, and you are seeing them at different time point intervals, whatever that speed of the sequencer is, maybe in milliseconds or basically in micro or nanoseconds this thing happens. and these cycles they continue, and the camera is taking the pictures, and finally when he compares the pictures at time point 1 and then at time point 2 and then at time point 3. So each time point represents a new nucleotide is being incorporated. So first nucleotide that was incorporated here was C, second in the same position was A, the third one was T, the fourth one was C and G and A, right? Oh, sorry, T. So C, A, T, C, G, T, and bottom ones are all C's. Similar is happening to the rest of those fragments, and this thing is being read or being written on files, on word files within the computers, and then finally when the sequencer returns them, we call these things as read fragments. So originally we call them as fragments. So when they are read, we call them as it should have been called as uh, readed, or I guess the second form is also read, so that's why they're called as read fragments, or they were just reduced to reads. So what we call them as reads. So there is this top read and there is this bottom read. So here we are seeing two reads. Individually it's a read. So two are reads or more are reads. So two read fragments or reads are here, right? So the top one is CAT, CGT. Normally it's not 6 bases like that simple here. They're like 100 bases or more than 100. They can be up to 400 plus 450 or so far. 454. Illumina goes from 50 to 250 bases, right? The maximum which we are getting in my seq sequencer is 250. Some of them they go like 150. High seq or high scan, that is very high speed. So that goes so as you decrease the fragments length, obviously, and since you are processing all those fragments in parallel, so that's increasing your speed, right? If there are the number of fragments, that means the number of spots you have or the positions that can be occupied here are less on that instrument, that sequencing instrument, and you are keeping the read lengths more, so obviously it will take more time. So this thing is also got famous as short read sequencing technology, right? So short read. And now in the third generation, we again wanted to have the longer reads. So rather than sequencing by senses, they adopted the real-time sequencing. In 454, what happens is when a nucleotide is added, a pyrophosphate molecule comes out, and then the system allows him to react with the other molecules, and finally it generates a light signal. So anytime you detect a light signal, here the light is of same color, not four different colors. So another strength in the algorithm of Illumina over 454 are similar methods, that here the light is of only one color. So how would you then you distinguish whether it's an A or C or T or G that has been incorporated? That's my question. So if it's only one color, so how can you distinguish that which base has been incorporated, whether it was A or T or C or G? How would you ensure that? Anyone? So, yeah. Someone is trying to speak. Go ahead, please. Vishal, maybe you can write. We can't hear you. So what happens is we do that in cycles. There is the chat window here, so you guys can write over there. So I can just keep an eye on the other screen. Voice is not clear. There is an echo. Is it so? That message was at 6:18. I could not see that. Sadia, you should also keep track of that.
Sir, it's better now.
Better now? Yeah. So what happens over here is that how would you ensure that this A has been incorporated here, or if there is an A on the opposite strand, so that means there should be an incorporation of T against it. If T is incorporated, what you have on the opposite strand, it should be an A, right? So how would you distinguish, because the light is of same color? So what they do is they wash those picotiter plates with different nucleotides, such that only one type of 1 out of 4 nucleotides they are washing those picotiter plates are on semiconductor chips in the next instrument, right? So then they take out those A's and then they put in the T's and then they take out those T's. So those places are those wells are those spots where you get that color. That means that nucleotide has been incorporated, and then you take that nucleotides out and then you bring the next one. So you are doing it in cycles, right? So every time you need to wash this thing 4 times at each base position. So at each sequencing reaction you need to do that thing 4 times. For some there should be A in it, for some they're gonna take T's, for some they're gonna take G's and C's. So you need to do that for cycles, and you need to wash them with one type of nucleotides, and then you need to remove that. Similar is happening here in OIN semiconductor chips. So you take that ball full of those fragments, the copies of that fragments, that monoclonally amplified fragments, and whenever a nucleotide is incorporated, there is a change in the pH. So that change in the pH is detected by sensors. They behave like mini pH meters. So those pH meters, they detect the change in the pH. So you wash these wells with different nucleotides, and you do it 4 times, like you do it with A's. So for example, this well number 1, that sensor detects there is a change in the pH. So that means that nucleotide, whatever it is, A's, has been incorporated here and here and here, just for example. And then you take out all those A's and then you wash it with T's. So they are incorporated here, here, here. Wherever there is a change in the pH, so that means that nucleotide has been incorporated. If A is incorporated, what you have on the opposite strand, T. If C is incorporated, what is on the opposite strand, G, and so on. Okay. So again, those cyclic thing is happening, but that's causing errors also. What if you have 2 T's? So over there's the double change in the pH, but 4 T's or 6 copies of T's at the same point. Over there, this thing is, it gives errors. We call them as homopolymer errors. These are the instruments of NextGen. 454 has this GS Flex and GS Juniors. OIN Torrent has OIN PGM. Solid has 5500 XL. HiSeq was the big machine, so mostly in our country we had seen this guy, the first one, and then this couple of machines are here in our Islamabad region, these OIN Torrent ones. But mostly we have Illumina, MySeq, right? HiSeq is the big one. Now there is gone. They had this updated HiSeq to HiScan, so that's even bigger than that. So this one is MySeq. That is the you can call it as benchtop machine. We have shared one video on human American gut project, right? So over there he's talking about MySeq. So it's actually he's selling MySeq. He's doing sort of advertisements for MySeq sequencers. So but over there that professor talks wonderful on that metagenomics or on that human gut project. So what happens in third generation is you don't need to amplify these things, because here you are not taking the pictures, but they came up with some instruments where you have this system of proteins. So like the extrinsic, what are you call them as, intrinsic and extrinsic, and there was the another ones, the integral proteins within the cell membrane. So they are trying to make something like that. Those transporter proteins, um, over there they have those conformations where the top one has a very small size pore in it, sorry. And then what happens over there is the flow of liquids, or the ions that's going through that, and that whenever those ions they are going, they are generating some electrical signals. And what happens is that you take the DNA single stranded molecule, right, and then you process that through this thing, and when it passes through one instrument called as adapter molecule, so over there you detect the change in that light signal. So different bases, the A's and T's and G's and C's, they change the polarity of those ions differently. That means they are giving us different electrical signals. So you can process the longer runs of the DNA. You don't need to process, you don't need to amplify them. There is no PCR here. So it's direct sequencing or real-time sequencing, right? And here you are generating the longer read fragments in kilobases rather than in base pairs. Here we are talking about thousands bases, so you can get like 30 kb or even more than that, right? So I think in our instrument which we had last month, this USB like thing, mini OIN, we generated the fragments up to 35 kilobases. So that's really, really long. So if you're doing a sequencing project, the assembly project, so these are the ones which are good, right? What is a sequencing assembly project? We'll talk about that in a moment. Sanger, first generation. Next was 454 Illumina and Solid and non-torrent, that is the NGS, next generation. And third one is also called as next generation sequencing, right? NNGS. So our third generation, so that's Helicose and PacBio and Oxford Nanopore. We did not talk about Helicose and PacBio, they are similar. So rather than calling it as next generation or NGS, some people argue that we should call it as HTS. So HTS stands for high throughput sequencing. Rather, so we should better call it as HTS. Right? Okay, so that's a comparative overview of Sanger. The read length what we get from Sanger is 400 to 900 bases, but they believe 700 is the best, somewhere in between. But you can go as low as 400 and as high as 900, or even you can go up till 1 kilo bases, 1000 bases or base pairs. In OIN semiconductor thing, we go up till 200 maximum. In pyrosequencing up till 700, right? Illumina 50 to 250 and Solid was like 50 to 85. Accuracy everyone claims 98 to 99.9%. Reads per run, it's not applicable because that depends upon how many uh, wells or how many, yeah, so that's a plate that has those wells. So normally we, which sequencer we had down there in our one of our labs, so that is retired now by the way, but that has 96 well plate. 96 multiplied by 1 kb maximum if we are going at maximum capacity. So that was generating 96 kilobases in one reaction, right? So that reaction costed us uh, more as compared to the other ones. So here that cost is heavy, but that thing has very high accuracy, so that's a gold standard. So once you have done with NGS, so specifically for that SNP experiments, SNP is where each nucleotide position you are presenting is important, so you confirm that with Sanger. So if you do that, obviously you can get that thing verified, and then you can publish it into some good journals, right? Ion torrent go takes like this time per run. We were here, I guess. So reads per run not applicable. Up to 5 million here in Ion semiconductor, 1 million in pyrosequencing, up to 3 billion. He is talking about the maximum capacity. So maximum capacity was that of the high-seq, so that is up to 3 billion, right? So Solid was like 1.2 to 1.4. Time per run 20 minutes to 3 hours, 2 hours, 24 hours, 1 to 10 days. If you are using the high scan or high seq at a maximum capacity, and it's all fully loaded, each well or each lane is occupied, so you are generating the 3 billion or whatever that maximum capacity is. I guess it's gone beyond 3 billion now in high scan. So you can take up to 2 weeks, like up to 10 days, 1 to 2 weeks for Solid. That's slower. Cost for 2400 per 1 million basis. That's $2,400 if you want to sequence 1 million bases, right? Here $1, here $10, here 5 cents to 15 cents. So that's more economical. That is again what I'm talking here is if you are running a sequencer of huge size like high seek or high scan, and you're running it at 4 capacity, then it is the cost is this, because you get more samples, you get huge data to process, so that cost is lower, right? But if you are cooking in a small pot, obviously your food quality is good, but that cost is more. But if you do it at a mega scale, obviously your cost per person reduces, right? Shadi wadi mein, in the marriages, you will use deg, right? You will not be using deg cheese, right? The small pots, dekche वगैरह आप use नहीं करोगे. So over there you can obviously अगर आप देगचों में पकाते हैं तो that food cost is increased, but quality is also increased. But here the trade-off is there. So that he's talking about the things that is having the maximum capacity, and then it is fully loaded. When you are running each and every nook and corner that is available, right? So you're not underutilizing that thing. So more you, if somebody is running at full capacity, obviously that will lower its cost. So what's happening in our region or country, or the people we see acquiring those sequencers, that was not a big problem. The problem is that the cost of running those sequencers is heavy because they are not having the enough data. Everyone buys a sequencer, so alone you cannot run it. At the University of Illinois, while I was doing my PhD project, I have to wait like 4 to 5 months to get my data because they put it in queue unless they have enough sequences. They were like, if you want to go real quick, we can process into MySeq or some other machines, but your cost will be heavier, right? So since we were dealing with 200 samples, so we waited for 5 to 6 months. That was worth spending our time. And I was a doctoral student with no experience in NGS data analysis. I was taking the courses similar to what you are taking today, right? This course, and from there I started, right, this thing. And then everything learned through experience. I'm still a learner, maybe a senior student than you guys, but that's how the things are going on. And then some of you who will go abroad, or some of you who are dealing these things in real, they might have the latest because we don't have that sequencing facility available here, right? And then another point is that while you are a student, you can enjoy your liberty and your freedom, and you can work and you can do trial and errors. But when you become a professor, obviously you manage the students, so your students are doing the work, but you are like a manager, right? So they are on the driving seat. So obviously the students for you, this is just a beginning, this is just a start, but I know that you will do wonderful few years from now. Advantages: long individual reads here in Sanger, less expensive in InTorrent. That's what they say, right? That's what they say, because but you can compare yourself. We guess this one is the cheapest. Long read size and fast in case of pyrosequencing. Potential for high sequencing yield depending upon the sequencer model, whether it's iSeq or MySeq or NextSeq. Now NextSeq is between MySeq and HiSeq, and then they came up with MiniSeq, even the smaller one. So the first one is MiniSeq, and then this MySeq, and then NextSeq, and then HiSeq. That seq word means sequencer, right? Or NGS sequencer. Okay. Disadvantages: more expensive and impractical for larger sequences. Homopolymer errors. If you have the runs of A's, how would you distinguish these are 6A's or 7A's here, right? Because the light is of same color. Runs are expensive. Homopolymer error. Two things here. It's expensive, $10, comparable to them, right? Here we are comparing these, we are not comparing the one in blue, that's another generation. We are just giving a comparison between Sanger and NGS, but within NGS we are talking about this comparisons where it's $10 per 1 million, but rest of them are $1 or 5 to 0.15, 5 cents to 15 cents. 0.13 means 13 cents, right? So we had rupees, and then we had paisa. You guys don't had that thing, but we have seen paisa also. So we had cents in our PKR, but now even PKR, its cost has been reduced to, nowadays I would say, what we had seen in our childhood. Now that thing has been reduced to 1 by 10, right? So that's inflation. So here we are talking about the American dollar. So 5 cents, though 5 cents will give you nothing, you cannot buy a candy over there, but we have seen the dimes and quarters like 25 cents. So that we use to do laundries, to put into those washing machines or dryers, or to put into those meters for parking our cars, on the roads and on campus. That was, by the way, quite expensive, right? So, and then we had equipments, they are expensive. So obviously Illumina, acquiring that technology is expensive. The other ones might give you the discount because this one is the true, the winning technology. So normally they don't offer us much discounts. Solid one, probably if it exists, it's gonna give you the free machine, but from where they're gonna earn that? Obviously, you need to run it, and once you need to run it, you need the reagents and the chemicals. Obviously, that costs. So we talked about the technologies and their comparisons, and here we talk about the applications of NGS. So if you're sequencing the DNA or the genome, you know that the genes are there within the within the chromosomes, right? And then if you have, multiple genes, are you if you are talking about the all genes or all whole DNA of an individual or a cell, that's called as genome, right? So genome. So the field becomes genomics. That word omics is coming from NGS thing. So we are living in omic era, and you might have seen the words ohm or omics. That word ICS is coming from, you had statistics, you had mathematics, right? Not in computer sciences, but in computer sciences we had that ICS degrees, intermediate in computer sciences in our country. I don't know, in America we don't used to call it intermediate, but they call it high school. So high school is up till 12th grade. So 11th and 12th is your high school, right? So that is the high school. ICS, intermediate in computer science. So obviously, that ICS word mean indicates that you are using computers here, you are using the techniques from maths and stats, and you are doing it at a big scale, at genome scale, so genomics, right? And then we see that sometimes that DNA is all good, all perfect, but within those proteins, we call them as histones, that part has, what do you call it is? Heterochromatin and euchromatin, right? And then we observed that the free part is expressing, but that part which is bound to those proteins, even the other proteins, they come and attach and bind it here. We call them as these sort of the ones which are from within, are that DNA sequence on which those proteins are binding. We call them as cis regulatory elements. Cis means on, that's the DNA part. Trans means those proteins which are coming from somewhere. These histones are synthesized somewhere else, but they come and attach here. So that's your trans part. So cis part and trans part. Mehakali, wanna enter the class. So let me head. Do you guys, the participants, has the ability to let the people in? If that, feel free to do that. So somebody has, yeah, I think we can do that. There is a voice note. Sorry, the chat box has something, so that was bothering me. Anyway, so what you do is, if you are taking that, uh, that DNA piece that has been attached with some external elements, or sometimes we see that DNA is perfectly all right, but some extra sequences or extra, molecules, they come and attach over there, like methyls or acetyls. We call this process as methylation or acetylation. So that methylated, or whatever the other thing is, acetyls groups, when they are attached, they change their gene expression, right? So that's called as the epigenetic things. The things are beyond genomic or genetic level. If it's with few genes or few, small DNA pieces, we call them as genetics, but that is the classical genetics. When you say genomics, so that means that huge or major scale. So epigenomics is you are doing it for, maybe one chromosome or some longer regions. So epigenomics is talking about those modifications or those external elements. And here again, let me clear one thing, that for us the DNA is important, that is attached to that protein part. We don't care about those proteins, we discard them, but we detect which part was being attacked by that protein and that was not expressed, or that has been regulated. This thing is called as regulation, right? So epigenomics deals with genetic regulations. And why epigenomics? Because epi means beyond or above. So it's beyond genomic level. So it's not from within genome, but some external elements are doing that. Mnemonics, microRNA are small elements. So those are the ones which attach to another RNA. Let me go here first. So DNA expresses and it makes RNA. So that product is called as transcript. DNA makes RNA. The process is called as transcription, right? So DNA makes RNA. The thing is called as transcription, right? And then this RNA is then translated into the protein. So which RNA is translated? Messenger RNA, mRNA. The other ones, transfer and ribosomal, we can detect them with these techniques, NGS with RNA sequencing, right? But here we are talking about that messenger RNA thing just for elaboration. MicroRNA is something that is complementary to those messenger RNA, so that blocks messenger RNA. So that means studying microRNA is also important. So that thing is called as mnemonics, right? Why microRNA are important? Because there is some gene that is cancer causing or oncogenes, and it has prepared its messenger RNA, and that is going to, prepare a protein, so that protein can cause that disease. So if you design a microRNA against that, so that will be real quickly targeting that at RNA level, because dealing the things at protein level then becomes complicated. Okay, right? So that is where you can do that early, you know, uh, cure or treatment. So this Mirnomics also got famous in COVID, right? When COVID came out, it's an RNA virus. So we had the medicines that were targeting that coronavirus, right? Or the other viruses, which are like retroviruses. So if you design those microRNAs, so that's called as microRNA therapeutics, right? So that's very interesting field to study. Transcriptomics is talking about messenger RNA, how much messenger RNA is produced, that is correlated with how much protein is produced. So rather than checking or detecting the proteins, which as I said complicated because one DNA or one gene makes multiple proteins through a process called as alternative splicing. So you get the alternative spliced variants, right? So you get multiple variants, that means multiple proteins. And then when those proteins are formed or when they are transported, they get modifications in them and they get changes in them, right? So studying those multiple proteins is really tough and difficult. So that's why this RNA expression studies, they came back to, rather than dealing these the things at protein level, the people decided to do it at RNA level. So RNA sequencing became famous, and then it got famous as, the overall field is called as transcriptomics, but this thing got famous as RNA-seq, right? So RNA sequencing. Before that, in between, they developed another technique called as microarray. So in microarray, we are not doing this sequencing, but we were detecting this RNA molecules through what we call it as hybridization. So we prepare something that is like this microRNA longer than that. That's called as probes. So those probes are single stranded. They are complementary to that RNA. So if you take a gene, you can determine a piece, and from that gene that is unique, that can only hybridize with the messenger RNA of that particular gene, and then we put a color tag, right, a fluorescent signal. When we talk about that RNA-seq, I will review microarray over there, and then there is a color change when that probe detects the relevant messenger RNA, when it becomes that RNA duplex. Normally they actually do it with cDNA, right, converting those both probe and both messenger RNA into DNA, so that DNA molecule is stable. RNA molecule is not that stable, right? So because of that, just let me get rid of this thing. So, so what happens over here is this field got emerged as transcriptomics. So transcriptomics, mnemonics, epigenomics, genomics, and metagenomics coming on next slide. So we are living in omics era, omics, right? The word om, right, is there, right? So yeah. So what you do over here is genomics, and within genomics, we see there is another four things which are written. So it says targeted sequencing. So sometimes we only detect some specific genes. So like in cancers, we observe that P53 is important, and then BRCA is important in breast cancers, and some other genes we just narrow down to suppose 20 to 30 genes, and we only sequence them. So in that case it becomes targeted sequencing. So targeted sequencing has this blue bar going upside down. Blue bar is current clinical utility. So maximum for targeted, whole exome is then in between, and whole genome has the minimum clinical utility, because clinicians need a real quick solution, right? So for them that targeted approach works well, rather than you just sequence that whole genome. Though in whole genome you have more information, right? That green bar represents your information content, right? So that thing is maximum in case of whole genome. So if you do that whole genome, you can, you have targeted all 25 or 27,000 genes of suppose human, and you can train multiple doctoral students on that. So that's a huge data which you are getting. So that means it has huge information content. Similarly, it has the huge cost. The experimental costs are those red bars. So they are, tapered up here. So minimum here, maximum here at the bottom. This thing is maximum here and minimum down, right? So tapers down below. This epigenomics and metanomics and transcriptomics, right now they're not that practiced much, but obviously that bar is not available here. But here you can see those two bars again. That targeted thing has the minimum information as well as the cost, but maximum at whole genome. So here he's talking about targeted bisulfite sequencing that is done to perform this or to identify the methylation sites. So the DNA pieces which are methylated, right? So those ones are then methylated nucleotides. So what you do is you take those methylated nucleotides and you treat them with bisulfite. So that bisulfite converts, I guess what was that, the one which was similar to uracil, I guess thymidines. So those thymidines are, so they are converted into inositol and then uracil. So the ones which are methylated, they are not converted. We'll talk about them when we talk about that targeted bisulfite sequencing in detail. And then the ones which are methylated, they will not be converted. So, and then when once those uracils are added to, are replaced, they replace the uracil is the nucleotide that is in RNA. The enzymes detect that part as something like RNA, and then they cleave that or they break that, but the methylated DNA survives. So in this way you can filter the methylated one, and then you can sequence, and then you can find which bases are methylated. So where are the methylated sites? ChIP-seq is chromatin immunoprecipitation and followed by sequencing. We'll see them in another slide and it will be clear. MEDIP-seq is something like ChIP-seq, but here we are capturing the methylated DNA. ChIP is chromatin immunoprecipitation, and MEDIP is methylated DNA immunoprecipitation followed by sequencing. That word seq means sequencing, and obviously we mean sequencing by which machines? NGS machines, next generation sequencers or third generation sequencers. And whole genome bisulfite, you can do it targeted or you can do it whole genome. In metanomics, you can check which parts of the DNA or which are the sources or genes which are preparing or which are making those microRNA, and then their identification or detection or editing is where you replace some of the bases on those microRNA, and if you replace them in the original DNA site, so you can damage or you can then control those. Like some of the microRNA may be bad because they are stopping that RNA for expression. So if that's some good gene, or maybe it's some tumor suppressor gene that was going to suppress the tumors. Which one is tumor suppressor gene? Anybody? The biggest one, the first gene that comes to your mind when you talk about cancers. Anyone?
P53.
Yes, so that P53 is basically P53 for protein and TP53 for the gene. So that one is where if it has some some microRNA is there and it is stopping that expression of that TP53, so we can design or we can edit, we can do some changes to the original gene's position on at some places or at some nucleotide positions to that microRNA. This thing is called as microRNA editing, so that this microRNA should not damage our target messenger RNA. Transcriptomics, we already talked about that. You can capture the gene expression, that is the messenger RNAs. You can capture the non-coding ones, the transfer and ribosomal and small nuclear or small nucleolar or whatever those species are. You can also detect the gene fusion detection. Some of the genes, they prepare RNA and then their RNAs, they give a chimeric product. Their proteins are coming from different genes. That means different RNA. They fuse with each other, or they somehow their product is then, and some of those amino acids are coming from one RNA, some coming from other. So that's the gene fusion thing. The overall product is called as chimera or chimeric RNA or protein. And then you have those splice analysis, like which alternative splice variants are abundant, right? So there is one study that says that within multiple RNA variants, there are the ones which are called as major variants or major ones, and then there are the ones called as minor ones. So some of the variants, they dominate their RNA pool for the same genes. So that is the study of alternative splice variants, right? Or at some conditions we observed that some of the variants, they are more and some of them are less. So which ones are condition specific alternative splice variants, right? And then the RNA editing, as we discussed here. The next slide, we talked about the central dogma of life. So DNA makes RNA, RNA makes proteins, and we already talked that our limitations are up till this level. We are not going to that level because that's the big thing. That's not my piece of cake. We already talked about that thing. If you are sequencing a genome or a DNA piece, or you are doing that for some part of the DNA which somebody else has not done before, you call it as de novo sequencing. De novo sequencing, right? So Human Genome Project was actually a de novo sequencing. So what you do is sequencing a novel genome for the first time requires specialized assembly of sequencing reads based on overlaps. So we need to, since while we were sequencing, we took the original part and we disassemble that. So now we disassemble, now we need to do what we call it as assemble them, or this thing is called as assembly. So genome assembly is part of de novo sequencing thing. You can also do transcriptome assembly, and you can make the longer transcripts, and then you can do something which you are doing, which we will be doing, what we see in resequencing. Resequencing is where you already have a sequence or already have a reference, like Human Genome Project gave us a reference sequence. So when we sequence, like when you know different countries started sequencing their own people, like that first Pakistani genome coming from ICCBS was a resequencing project. So they took those sequencing reads, right, and then they aligned those sequencing reads to the data, to the original reference that was there on that was produced by this Human Genome Project called as HG. HG stands for Human Genome, and then it has different variants. So it has been gone from 1 through right now we see variant 38, HG38, but they aligned I'm sorry, they aligned that with HG19, 19, right? So 19 or 19, and then from 19 the number jumped to 38. There was nothing like HG20 or 21 or 22. So they aligned with HG19. If you want to take that same data and you can get the updated assembly like HG38, and when you align with them, you might get some results that may be different. That is, one of our MS student did that, right? But we did not publish because obviously, most of the things were the same which they had. So we had very little extra information on top of it. So resequencing is where something is already sequenced. So you sequence your genome because assembly is expensive computationally as well as economically, right? So you need more money, you need more power, you need to have that whole genome in multiple copies. So over there, you need to be dead sure about your genomic assemblies, right? So assembly project is expensive, resequencing is easier. You can just compare with the reference which is already there. So you can find the similarities and differences. So only, I guess, 1 to 2% different exists between different human races. The people within one community maybe differ from each other at 0.2%, right? So that's very, very little. So there are 3.2 billion bases, right? But you can get those differences like maybe at how many, maybe, some 80,000 or 100,000 positions. And then you take those positions and again you narrow them to few genes, and then you interpret them. So finally we realize that maybe we are different in adaptations to our foods, our, our environment. So those things are maybe different, but most of the things are the same, right? Okay. So resequencing is where you are resequencing what's already sequenced. You are just comparing with that. So that's resequencing, right? So who is at the door? Sadia. Somebody is keeping track of the participants, because that thing then disturbs my flow of thoughts, and then obviously your flow of what you are listening, processing on your brains. So maybe somebody should take control of that entry and exit thing. I mean, you don't need to control exit, anybody can exit, but I don't know, somehow settings I try to change them because somebody need to keep them in targeted sequencing or targeted resequencing. What you do is you can just take some pieces or portions. The generated sequences align to reference sequence and mine for SNPs, single nucleotide polymorphisms. I'm sorry. We also call them as variants, single nucleotide variants. So SNPs or SNVs. SNV is, it's if it's a new thing. SNP is maybe in that population you are seeing that variant in more frequency. So then it becomes single nucleotide polymorphisms. So SNV. I can't hear you. Is there a question? Maybe you can type in the chat window and I can see that who's out there. I maybe just mic switched on and you're talking to somebody else. Right, so the generated sequences is aligned to a reference and mined for SNP. So I was telling you the difference between CNVs or SNVs. CNVs is copy number variants, so you can have multiple copies of some genomic regions within different individuals. So that's CNVs, copy number variants, right? So multiple copies. Some regions are present in multiple copies. Anything that comes to your mind, those copy number variants. Right, so those may be your markers. ALU repeats are, what you call them as SSRs and similar to that, right? So that is where you get the multiple copies and it is individual specific, right? So VNTRs, for instance, their pattern can help detect a person, right? That's what you do on gel electrophoresis thing. But in gel electrophoresis, the classical techniques where we observed that the populations, like somebody from our Gilgit Skardu area pointed out that over there this technique is not working. So that's forensic guy told us that we need NGS early as possible. So that was his idea to implement that NGS rather than that original VNTR based methods where you cannot you're getting those uh, SNPs, such that even that this NGS thing can detect a one base difference between two individuals, but that classical you know uh, gel electrophoresis are those techniques where you're looking for the bands that's not have the enough power to detect, right? So it's losing its power. So genome sequencing is where we talked about DNA sequencing. What you do next is you do sequencing and then you do quality control and then you do assembly. Now we are moving towards data analysis. And once you assemble a genome or a chromosome or some region of the DNA, some big part from those fragments, those reads, we identify protein coding genes and non-coding regions in it. That thing is called as annotation, right? So something similar to you get some script in some language and then you hire somebody and then he studies that and then he tells you that this is where the actual message starts. So that thing is then decoded. So that decoding thing is what we call here as annotation, right? So it is the term that is coming from linguistics, languages, right? So understanding what the message is all about. So identify where are the protein coding regions and what we have in it, where are the non-coding regions, where are the regions which are expressed, where are the regions which are not expressed, where are the exons and introns. Similarly, you can grasp variations within the genomes. That's also is here in genome sequencing, right? When we sequenced human genome, that was a universal thing. But when we sequenced Pakistani individual's genome, right, from ICCBS, I mean, that is where this ICCBS got a pace in this sequencing thing. Let me give you the hint. So that was the genome was coming from one renowned scientist, and he was kind enough later on to give us this Jamilur Rahman Center for Genome Research, right, at ICCBS, right? So, and then they compared it with Indian genome or with some other genomes, and then they discussed like how much we are similar to Indians. But obviously, your sample size was low, but that was the first effort, and obviously, it meant a lot. I guess they published in I believe 2014 or 2015. Expensive a full genome rather than targeted. So targeted is easy. You are taking just a small piece. So obviously it's not that expensive, right? Human Genome Project is, or human genome, you can get it from UCSC. You just click and then it will open up that UCSC genome browser. So over there you can then study and it will give you a file. So that has all those 24 chromosomes separating X and Y from each other. So they are total 24, right? We say 23 pairs, but what about if there is no pair? So we had in males, we have 23 and 24. So 23 is X, 24 is Y. So all 24, their sequence, and then some of the sequences are not assembled. So they are just what we call them as additional regions. So they also put them, right? Yeah, additional fragments, maybe in the shape of what we call them as contigs, because reads, when you put together and you assemble something out of it, that read is then not converted into reads put together make contigs. Contigs put together make scaffolds. Scaffolds are then put together into chromosomes. We talk about these things when we talk about genome sequencing. So in exome, you are actually capturing the exons. So sometimes it's partial. Exons are only the coding regions. Okay, you are not talking about. So here you are talking about the exons that are already identified. Okay, if you want to identify some exons that was not pre-identified, then you need to go for genome. Okay, so exomes. So you can detect genome-wide variations in exons and adjacent splice site. Cost efficient, so mostly people are working in human genetics, like Miller or Freeman or Kabuki. I don't know what the hell these are. I mean, the human genome people are here. They can tell us ARVC and ataxia. These are the genetic diseases mostly, right? So yeah, so over there they are using it. So mostly the human genome, the people who are working with the human genetics or genomics, so they know what I'm talking about. Or the people who are working on those diseases, even the medical community, they are familiar with that. So exome is very much popular in them. Transcriptomics is, here is this 1,000 genome project. So where they are keeping track of variations coming from, originally they started with 1,000 genomes. So while they were doing that, I heard, and my source is Dr. Kamran Azim, the author of that paper or that research project, a wonderful person, must appreciate. So yeah, so that he did this first Pakistani genome sequencing. Right now is in Maju, Karachi. He left ICCBS, so he's there. So as a dean or pro vice chancellor, I think. So Dr. Saab told me that at that time they were giving us the people who were volunteers the free flights or free tickets to US, come and donate your genome. So they were interested to collect 1,000 genomes from all over the world. Later on, it became 100,000, and now I believe it's 1 million genomes. And obviously, they are not offering those air tickets now, because now people are sequencing these things all over the world and submitting those sequences to them. So they had a huge samples within that. So if you go there, last time we went was few years back, when one of our student, he was interested to work on comparative genomes from different regions like Pashtun versus Punjabi versus Balochis or Sindhis, and finally we observed that in some of them, in comparison to each other, right, depending upon their dietary habits, we observed those snips, right? So people like Lahori Punjabis are fond of food, but obviously they are at risk of getting those cardiac diseases or diabetes, or so we get more snips on those regions, right? So hey, people, just think about and reconsider what you are eating, right? So especially the people of my age, now we cannot eat anything, and we cannot no, I'm sorry, not everything, right? Not every time, right? In transcriptomics, where we are capturing the genes which are expressed, so here we are just looking into a reference. The reference is, for example, HG19 or 38. That's this white line over here. I'm sorry, let me elaborate that. So you might have seen these pictures in the papers, and these are the individual read fragments which are aligned to those reference. So in exomes or in transcriptomics, we need to do that alignment to the reference. So that reference already exists, so we align those reads, and then you can see the similarities and the differences. And then we zoom in, and this cloud is representing those individual reads, where you get thick cloud, that means maximum coverage or maximum highly expressed, and then the regions where that those read fragments are less, so that is where you see less reads are mapping. Maybe there is some deletion towards your sample, though that thing is there in reference, but few reads are aligning. Obviously we zoomed in, but actually in real life that region was really small or negligible as compared to those regions, right? So within that highly dense region, so we are picking up one position, and we are seeing that in the reference it was G here, but on the reads or fragments which are mapping here, the read fragments are the sequence fragments that are coming from sequencers. We call them as reads. So here we are seeing those reads, right? The red ones are pointed this way, the those gray ones are pointed forward, and some of them they align in reverse direction. We get their reverse complement and align them. We'll talk about that when we talk about these things in detail. So here at this position on reference there was Z, but on your sample which you sequenced, that had C. So that means this guy had a single nucleotide variant here. So that may be a potential variant. It says putative variant, so there would be a variant, and then we test it, right? And then we verify that before declaring the actual variant. Maybe it's a sequencing error or something like a PCR error. So those PCR things are then they are not like multiplying those fragments, the same fragments, and getting their multiple copies sometimes give us the PCR duplicates. We call them as duplicates. Though we had the duplicate regions within our genome or DNA, since we had two copies of the chromosomes, that means two copies of the genes, and obviously there are the regions which are repetitive regions, or the normal multiple copies of some sequences are already there, but we call them as orthologs or paralogs. Yeah, paralogs within the same species. So we believe that according to Human Genome Project, more than 40% of our genome is repetitive regions, right? So that's a potential candidate to be a SNP, single nucleotide variant, or SNV or SNP. Then we look into that population. If that variant is already there, reported in that population, then it's not variant, it's P, it's polymorphism, right? So variants are given more scores than SNVs are good. I mean, good in sense that if you detect them and you are a doctoral student, so obviously that can lead you to some novel discovery, right? But something which already existed, and its frequency is, I guess, more than, what was that, 5%. So if it is more than 5% within that particular population, right? So in that case, it becomes a SNP rather than SNV, right? It becomes SNP, right? So transcriptomics detects the transcriptome or RNA. Repertoire means collection of RNA repertoire. Gene expression studies like mRNA studies, what we call them as RNA-seq, right? Plus there are multiple forms, isoforms, editing products, obviously where those editing thing is done in reality, right? We can study them. Not coding RNA we can also capture, like ribosomal transfer, ncRNA, small RNAs. Meronomics detects microRNAs. So again, that's also the within transcriptomics, and epigenomics detects what we call it as meronomics detects microRNA. These are small non-coding. Maximum length is 22 nucleotides. So over there, you can get a sequencer that is going, that is, giving you a small size read fragment, something like Illumina's. Some machines are giving us 50 nucleotides. So if you are doing it on high-seq or high-scan, you are wasting your resources, so that will increase its cost. So something like 22 or 23 bases long read fragments are good enough. MicroRNA are involved in genetic regulation, post-transcriptional regulation we call it as. Okay, so there is transcription, that is DNA makes RNA, and post-transcriptional means, so after the RNA is formed, there are some changes, or if after, while it's transportation, there are some things which are happens. And remember, there are also post translational modifications, PTMs, right? So that is when those proteins are formed. So some of the extra, things which are added to those proteins, so they can change their expression, or they change their function, right? So that thing is called as regulation, gene regulation. So people who are taking the gene regulation courses, they might have covered these things. Epigenomics is in RNA sequencing. What you do is you capture RNA, messenger RNA, and messenger RNA is different from rest of the RNA. Which RNA species, by the way, maximum or more in all RNA, types? In RNA types, which RNA is most frequent? What are your view?
Ribosomal RNA.
Ribosomal. So ribosomal does not have this poly A thing. So that is the difference between messenger RNA and rest of them. So while you are capturing messenger RNA, so one of the technique insists that you can get those oligo DTs, deoxy nucleotides, and then obviously you are taking that messenger RNA and you are converted to cDNA, right? And then that cDNA, you capture them with the help of poly A tail method, right? And then you convert them and you make the cDNA, and then you break into the fragments. The rest of the things are the same. So poly A tail method is the most famous, but sometimes some paper came out and they said that that method is not good. What if the tail is broken, right? What if the DNA is degraded? You put it into, like in the human subjects, they used to get it from cadavers, or you used to store it in formalin, and you store it for long, so some changes happen. So over there, the other methods, they also came out, right? Epigenomics is where you are capturing the things which are beyond genomic or DNA level. So that's your DNA sequence. Here we are actually trying to see where those proteins are attached to DNA. We actually want their binding sites, right? So ChIP-seq is chromatin immunoprecipitation. Uh, have you studied immunology? What is immunoprecipitation? Can anyone? I can take a couple of sips of water. So just talk about immunoprecipitation. What is immunity? What is antibody? What is antigen?
The antigen is a protein, and antibody is another protein-like substance that attaches to the antigen, and it is specific to the antigen. The antibody is specific to that thing. And in immunoprecipitation, we use this. In immunofluorescence, we use these two things in order to stain something.
Yeah, yeah, excellent, excellent. Thank you so much. So what you do over here is you want to know what the sequence of the binding, where are the binding sites of those proteins. So that means what is the sequence that is here. So what you do is you take those proteins as antigens and you design antibodies against them so that those antibodies bring in those proteins, they precipitate them. As a result, the DNA which is attached to them also comes out, right? And then what you do is you remove those proteins, and now you had that regions, only regions where those proteins were being added or where they were attaching, so the binding sites. And then you sequence them through sequencer, and then you align them to the reference chromosomes, or you get that for example from chromosome number 22. So you align them on that particular chromosome, and then you can get the regions where those binding sites are, right? So those binding sites we are actually focusing where those proteins they go and attach, where you need to work, right? With the suppose some protein is coming and attaching, and that is harm for the body, it's causing some disease. So maybe you can change the binding site, right? And do not allow or do not let that protein sit in there. If it is the region that is causing some disease, then you can obviously facilitate that protein to bind over there, right? So drug designing in which this thing is important. Okay. Metagenomics. Metagenome means somebody was asking about it. So here it is. You get mixture of genomes. So we are seeing the threads of different colors. So ये मतंजन सा बना हुआ है यहाँ पे. We fragment them. We extract the DNA. You can take soil sample and then you extract the DNA, and that has mixture of the DNA. Obviously, you will not put the soil in the sequencers, okay, or the stool samples or anything else. So what you're going to do is you need to extract the DNA, and you believe that there are a mixture of genome here. That mixture of genome is called as metagenome. Meta means many, right? So many genomes are here. And then you fragment them, and obviously you get that Matangan thing out. And then you align, you do that assembly thing, and you can then identify, hey, we got genome A coming from bacteria number 1, and genome B, bacteria 2, and genome C, right? And then you can also now there you in whole genome metagenomics, this is called as whole genome, right? So in whole genome you identify those genomes, and then you sequence them, and then you align, you assemble them, right, or align them to even their reference, and then you can also detect. But here your objective is what genes are there, right, how it's causing that antimicrobial resistance. Nowadays AMR, antimicrobial resistance, is the thing that's gaining popularity. In one of our event coming at mid-November, they gave me the target to speak on AMR using NGS. So one of the, I mean, this Illumina is organizing a conference. Maybe we can share the details with you at Lahore in Pearl Continental Hotel, PC Lahore, on 16th of November, right? So we are having uh, different people who've been working in different projects. Though, like my, I work very just a couple of projects in microbiology, but mostly we do with the human subjects or the plants, obviously. But acha, what is the difference between whole genome metagenomics and there is another called as 16S? In 16S, we know that that region 16S, uh, it's coming from that S stands for Swedberg units. When we talked about ribosomes, we had the smaller subunit as well as the larger subunit. So in bacteria, one of them is of size 16. So 16 Swedberg called as 16S, right? What size we had, I think we covered in one of our cell biology course some 20 years back. So what we had in the eukaryotes, right? And then when it fits in, the total size is then reduced to just one or two further. But in bacteria, one of them is 16S. So within that 16S, we have a region that is different in different bacterial, say different bacterial species or phylums or classes, right? So we can just detect which bacteria are there. So who is out there or who's out there? If that is your question, bacteria A, B, C, D, E, and F. So in that case, you can do 16S metagenomics. But in whole genome metagenomics, you can also detect their genes and what they are doing and what their sequences are. Which genes are, how many antimicrobial genes are there in suppose Salmonella versus, something else. Right now, I got this H. pylori, by the way. We talked about that, I think, in the previous lecture. I just put those slides up, but I forgot to give you the updated one or forgot to save it. So we already discussed that. I hope you might have seen those databases where we have those NGS data, right? This gene expression one was our favorite, and most of the students they work with it, where you can get that RNA sequencing data when we do that RNA-seq studies. So most of you would love to do that. My favorite is also RNA-seq because my PhD was in RNA-seq. The technique started in 2008 with a research paper from the author in one of the famous scientists, Professor Barbara Wold, in her lab, and his name was Ali Murtazavi. Ali Murtazavi is an Iranian by origin, so he was a postdoc there. So he did a wonderful job in 2008, and then this technique then replaced, mostly, put microarray towards backseat, and this RNA-seq was, and still it is the, I guess, more of the 90% of the NGS projects all over the world are in RNA sequencing, right? At that time, and now maybe it's like 80 or 70%. And similarly, exome got famous. Genome is mostly with the smaller genomes like bacteria, etc. The microbiology people deal with the genomes, the smaller ones, and obviously there are much variations, and that speciation thing is frequent. More variabilities are coming in viruses. So obviously over there, you would love to do a genome assembly project, but doing a genome assembly project with humans or some eukaryotes is quite, laborious task. You have to be patient, right? That might delay your PhD. Depending upon which genome, up till that time, somebody in microbiology would publish 10 genomes, right? So that's what is the downside. But obviously, if you are working with the big things, you should have a big heart, right? So that is GEO, gene expression omnibus. We just went to one of the records and we can see it's mus musculus mouse. It's dicer knockout NSCLC, non क्या था बाजी, small cell, non small cell lung carcinoma. RNA-seq and microRNA-seq. So they pooled both RNA and microRNA of mouse, and that was they were the knockouts. Dicer is an enzyme, I think, that is there at the microRNA prep stage, right, which processes them, right? So they knock out those mice for this that dicer protein, probably to knock out or probably to knock down their microRNA and then its effect on the RNA. So if you knock out that dicer thing, obviously your RNA, microRNA species would be changed. And what is the effect on RNA-seq? Suppose this is just a record. Experimental type is expression profiling by high throughput. That means it's an RNA-seq study. Also non-coding, because this microRNA is also here, MIRSEC. So non-coding RNA and a microRNA, and then Dasser knockout. Again, that summary Since we this study was contributed by these people and this was the paper, you just click here on this record, the paper will open and you can see what those people have done. They did it at MIT, Massachusetts Institute of Technology. CD Chen is the name of the author, and this slide I took in April 24, 2014, or maybe that record was submitted there in 2014, right? So that is where when you click to the next page, that's their series ID. You click and then you can get its sequence data here. It's called as FTP, SRP, or SRP041. That is where you can get their RNA sequencing data. When you when you click this file, series matrix, you get an Excel file where you can have that expression values um, in counts, right? And that platform is also written over here, Illumina HiSeq 2000. That's what was used. So I think now most of you are understanding what I'm talking about. That is this work, this study, right? So within the study, you can also get that record down there. So here you can click or you can go to GEO and put that GSC 3494 and you can get that data. Suppose they have presented mouse, the genes which are related to diabetes or somebody else. What was that? The NSLC. Maybe you can take some other set of the genes and find something different or find something interesting, and that can help you do your thesis work for free, right? 1,000 genome can gives you the data. Similarly, encyclopedia of DNA element, that project is in code, that also gives you that SNPs data. NHLBI, National Heart Lung Blood Institute, the people working on blood diseases, right, our heart and lung, they can get the data from here, which is already there. These figures are old, by the way. No, they will be having so many uh, like so much data available to them. De novo mutations in autism, that's their human microbiome project that you might be interested here. So to characterize microbial communities found at multiple human body sites and to look for correlation between the changes in the microbiome at different body sites, right, and human health. Collected samples from 242 healthy US volunteers and tissues from 15 body sites, 15 in men and 18 in women, right? And more than 200 researchers from over 80 research institute they became, and then later on they expanded to even the people in Pakistan, like some of our known people, or maybe they used to came in our trainings. They told us they get funding from that American Gut Project, and then it extended to overall human microbiome project, and they were giving funding, and you can go and sequence the people in your community, or you can get the representative sequences all over the country, and you can study what our microbiome contains, right? Ever heard of probiotics? Anybody? So probiotics are, if you take all the microbial diversity of some region, then you can design or develop some additional medicines, or the pills where they can have those microbes. So when somebody travels to USA, for example, so those probiotics might help them, settle down and digest their local food. And obviously, waterborne diseases are more, you travel somewhere and they say कि वो क्या बोलते हैं जी, पानी रास आ गया या नहीं आ गया. So that water which suits you or it does not suit you, it depends upon the microbial diversity within your body. If you are lacking some of them, maybe in future you will get those pills, those capsules at airports, right, before you exit and eat something over there. So next lecture we can talk about the data analysis. I guess most of you are now familiar with what's going to happen here in this course, but now we move on to the data analysis, and then in the second lecture in this week, and obviously the future lectures are all about data analysis. So you would love and enjoy that. So I guess in this week, Sadia might gonna take another extra session if you are available or if you agree, or maybe we can do another online session probably on the weekends if that suits you, right? And then we can have those tools installed on your laptops, and you can do all that thing and manage them on your laptops. We'll have the small data sets coming from our real data sets, not big enough to bother you. You don't need to have big workstations. You can manage on your laptops, which you are using for right now for your education or even write-ups, writing purposes or teaching purposes, right? So we'll be managing that. So with this, I conclude here in as far as today's lecture is concerned. If you have any questions or any comments, or feel free to discuss the things with each other or do some networking, right? Why don't you put your name and address on WhatsApp group we have, or on the chat window, or maybe you can post your questions over there, right? And we can discuss them. Feel free to speak if you want to. Okay, so that's all from today's session. Questions, concerns, queries, you're more than welcome to participate. Just ask. Is not clear, right? And that WhatsApp group is also our discussion forum. Some of the people are really asking very interesting questions, right? And we comment sometimes, and sometimes we had the other experts, right? Maybe you can tell us better if you are in microbiology about metagenomics, because we are mainly the data analyst, the biologist turned bioinformatician, right? So that was my background. Anyways, so yeah. So if in case there is no question, I would let you go. Recording will be available very soon, right? So here I can end the meeting. Should I end it? Thank you so much, Mariam, for appreciation. Yes? No? No questions? Thank you, Vishal.